So I'm recording this because we had some issues on Tuesday with doing the nucleotide sequence alignment with DNA Subway. It looks like some of their settings have changed, so there is a way to do this, and I'm just going to walk you through it. So if these are, let's say you're going to do this with HisG and HisD sequences. So let's say we wanted to do this with our HisD sequences. So we have our HisD wild type sequence here. And remember, this has to be in FASTA format, so make sure you get rid of the spaces here so that you can tell the difference between your sequences. You have the HisD here, and then let's say this is our sample, your dideoxy sequencing results. So what you can do is first go to DNA Subway, and you can either log in or enter as a guest. And you're going to do project type DNA, whatever your project title is. The key here is you have to start by only entering one sequence. For some reason now it doesn't allow us to enter multiple sequences. So we're going to copy this first wild type sequence. We're going to put it just like this. And we're going to click continue. And it's going to bring us to our screen here so you can see the sequence has been added. To add the additional sequences, we have to do it one at a time. So what you can do is click on Upload Data, copy the second sequence, like so, and paste it in this box here which says Enter Sequences in FASTA format. So I've copied that sequence. Click Add Sequences. And then the same thing with the third sequence. Now if you click on Sequence Viewer, you'll see all three sequences are in here. Our sequencing results for the revertant sample, the HisD mutant sequence, and the wild type sequence. So I'll X out of that. You can now use sequence trimmer to cut down the ends on either side. So you've trimmed off the ends on your sequencing results. If you want, you could click on Blast and just check that your sequencing results are in fact Salmonella. So we click Blast here. You'll see that these are our sequencing results. In fact, the top hit is Salmonella, so that's good. And the last thing that we want to do is click on muscle here. So this is the same muscle that we used for DNA barcoding. We click on muscle and then view. This is going to show the three sequences next to each other. It's going to line them. You can see that, remember, the dark bars here mean that there's no sequence. The light gray bars mean that they're identical sequences. So since the revertant sample, we've only amplified part of the gene, only part of it is going to match up down at the bottom here. We're interested in finding the mutations that are relevant to what we're looking for here. And we can always go back to our lab manual and find approximately where those would be. So for HisD, that mutation is at nucleotide 893. So you can see right here, 893. So we're going to look for approximately nucleotide 893 in our alignment viewer. And you can see the nucleotide numbers are up here, so it's going to be probably these colored bars right here. It's a little easier to click on the ATCG to look at it, and then we're going to look for approximately nucleotide 893 up top here. And you can see here 900, so if you were to count these off, this is nucleotide 893 here. And you can see with the wild type, there's a C here. With the mutant, there is not. A dash means that there is not a nucleotide there. So this is as we expect because this mutation is a deletion of one cytosine in the mutant. So this is good. Then we want to look at our revertant sample and see what happened with our revertant sample. So if we compare the revertant to the mutant, you can see the revertant still has this C. So because our revertant came from the mutant, there were additional mutations in the mutant that led to our revertant sample. The revertant still has a dash here, meaning it still lacks this C, but there are 2 other dashes down here. So what this means is that in our revertant sample, there were 2 additional single nucleotide deletions right here, a deletion of this G and this C, and if we add up the total number of deletions in our revertant sample here, you can see there were 3 total deletions, and we know that if you delete 3 nucleotides, that's going to put the sequence back in frame because codons are in sets of 3. So at this point on, from here on, the amino acid sequence is going to be the same as the wild type. So what you can do is you can actually look at what would be the effect of the amino acid sequence in this region right here by going back to your muscle alignment. So you want to kind of compare your nucleotide alignment with your muscle alignment, and your muscle alignment, like we're doing right here, is going to tell you what was the actual nucleotide change. So here in your revertant, there was 2 additional single nucleotide deletions. When you put these nucleotide sequences into XPASY and translate them, and then you align them in the muscle program like we did in lab, then you can see what is the actual change in the amino acid sequences. And and that's the extent that we need to look at for what was happening here. I did not realize at the time you can also still use this muscle program for DNA. So if you click on muscle, you can put in all 3 samples here. You can paste all 3 samples in here. And if you click submit, it will align the nucleotide sequences. It's not as easy because it doesn't give you the number of the location of the nucleotides here. So you would have to go down and specifically look for the mutation where you think it might be. This is, just from previously counting off, this is the original mutation in the mutant. You can see there's a loss of this C, so there's a C in a wild type, and the loss of the C in the mutant, and still lost in the revertant. And then here are the 2 additional deletions. So no matter what your revertant sample is, you can do this in either DNA subway or in muscle. You can do this analysis the exact same way, and what I would like you to do is try to identify what those additional mutations are. Now this is with the HISD. The HISG is going to be very simple because it's just everyone had a simple substitution mutation. So if you do this alignment with your HISG genes in subway, DNA subway, or in muscle, what you're simply going to see is the single nucleotide change, either at the original location. So remember with the HISG, we have this single nucleotide T to C substitution. You will either see the original substitution back to C or back to T, or some people saw a different substitution that led to this instead being a serine. So you would be able to easily see this in DNA subway or muscle as well. If you have any questions about how to do this, please contact me. Come see me. It's relatively simple, and this is the analysis that I'd like you to do for the lab report.